Okay, I think we'll get started. So welcome everybody to another of our series of machine learning and physical sciences Nexus talks. Before I introduce today's speaker, I'd like to remind folks that we have a big meeting on machine learning and physical sciences on Monday, October the 26th. It runs most of the day. You can get a link to it on our Nexus website. And that'll be instead of the normally scheduled talk that would be on the Tuesday. So that's on Monday, 2 weeks from today. Okay, so but today, it's a great pleasure to introduce Carter Butts, who surprisingly is from the School of Sociology. If you go take a look at his website, you'll see that he's involved in many things, including lots of machine learning and statistics. And what he seems to be really an expert in is joining things, right? Because he's a member of how many different institutes are you a member of, Carter?
I lost count at some point.
Yeah, I think
probably stochastic anyway.
Right, 5 or 6, but certainly the Institute for Mathematical and Behavioral Sciences and Cal IT2, and then about 4 or 5 different departments. So Carter is truly multidisciplinary. A lot of his work is in behavioral studies, but always bringing, a fully, let's say, quantitative view on things and using lots and lots of machine learning and statistical methods. So, and Carter got his PhD from the home of lots of machine learning, Carnegie Mellon University, and a BS from Duke University. Okay, so Carter, take it away, please.
All right. Well, now we do the eternal question whether technology will work. So let's see. All right, good. All right, well, thanks for the introduction, and thanks to all of you for tuning in today. What I'm going to be talking about today is some work that I and my collaborators have been doing, dealing with an issue that is, I think, kind of increasingly common in the physical sciences as well as other fields, which is dealing with the enormous surge in simulation data that has been made possible by the advances in simulating molecular and other systems. So, for a long time, people struggled with the question of, can you model things? Well, now you have the problem of, you have modeled things, you have a whole lot of trajectories, and you don't know what to do with them. And I think the machine learning has a lot to say about how we can try to extract useful information from that kind of information. So just give a very brief roadmap of where I'm going today. I'm going to first say a very brief word on kernel methods. And then we'll talk about 3 different applications and then of that, and then wrap it up. So I know this is a diverse audience, and some of you are doubtless experts in this area. Others of you may be just learning about it. So I'm not going to have time to go into enormous depth about kernel learning, but at least for those who are new to it, I'm going to say at least a few words about what it's all about. So it's a big area. There's a lot of work going back several decades. But I like to think of kernel learning as something that fuses ideas from several different areas. And so on one hand, we can think of kernel learning as a way of generalizing ideas, classical ideas from linear subspace methods and statistics, things like least squares, principal components, and the like. Linear learning ideas from classical machine learning, so linear perceptrons, support vector machines, things like that, and classical ideas from multivariate analysis, so similarity-based clustering and scaling methods. So those kinds of ideas, it sort of generalizes them and combines them with ideas from basis set methods and functional approximation, ideas that, of course, are of wide currency and long-standing use in the physical sciences as well. And of course, it encompasses other things as well. For instance, a lot of ideas in Bayesian nonparametrics, especially related to Gaussian processes that are connected with kernel learning. So what kernel learning kind of does, in a sense, is it takes all these ideas and it gives a fairly unified way to think about think about them. And as you might expect, sort of the kernel of the kernel idea is this thing called a kernel. So what's a kernel? Well, we could spend a long time on that, and we don't have time to do it. So I'm going to give a very, very shallow sort of description. I have to put a slide up with math on it because otherwise I'll revoke my math license. So I will put those up, although we won't talk about all of it in detail. But I do want to say at least a little bit about what a kernel function is. Okay, and so the idea of a kernel function is relatively straightforward. So we start with the idea that we're interested in studying some system, lives in some space. We can think about that as defined by some set S. I'm not going to make any assumptions about this S. It could be some nice, well-behaved thing like Euclidean space, but it could be a set of documents. It could be a space of molecular structures. It could be anything. So I'm not going to assume that it's particularly nice. But I am going to assume that we can define some vector-valued function phi on S that, so again, takes S into some n-dimensional reals. And in fact, we can even generalize that and let n go to infinity if we want to. But for now, let's just think about this as just something that takes an element from S, an element of the inputs, and produces a vector. And we sometimes call that a feature map. And so we call the things that phi function produces features. Of course, you can think of this as also defining a basis, which turns out to be an important way to think of it later. So, given that we have some way to map the elements of S into the reals, we now define the function that we really care about here, which is a function we're going to call k. And what it does, it takes pairs of elements in the input space, that is in S, and it maps them the real numbers in such a way that you can write down that function as a dot product with respect to the basis defined by phi. That is, if I take X and I take Y and I apply phi to them and I produce the feature vectors that go with them, and I take the dot product of those feature vectors, okay, that's what k computes. And anything that does that is a kernel function. And that's what a kernel function is, which at one level may be kind of disappointing. It doesn't seem like all that much, but it turns out to be a really useful idea. And part of the why, I mean, we'll talk about why in a minute, but one reason to kind of hint at why that's useful is because k doesn't have to be written literally like that, like I wrote it. So of course we could implement k by just literally applying phi to its inputs and taking the dot product. But in many cases, we can have a function that does that computation implicitly or tacitly. And this turns out to be really useful because we may have a situation where that feature vector is very, very large, or where it's very hard to compute, or we don't know how to compute it, or the like. And so we can use k as a tool for computing inner products in a sort of abstract way. In practice, we also apply some other constraints on k. In particular, we usually assume that it's at least conditionally positive semi-definite. Intuitively, what that does is it makes k, the kernel function, act like a similarity function. Okay, so it implies a weak kind of transitivity. And that turns out to be very useful as well because, as we'll see, we can think about using k as a way to talk in a very general manner about objects being more or less similar with respect to some set of features. And in particular, one of many ways to think about a dot product, right, is that it's a kind of similarity function, right? You could think about it as a function that gives you the cosine of the angle between two vectors scaled by the vector lengths. So you could think about k as taking the similarity of its inputs with respect to this basis associated with phi. And that turns out to be, again, a very useful idea when you want to compare things. And you can do a lot of other things with that as well. In particular, there are a large class of function spaces, reproducing kernel Hilbert spaces, that you can implement by kernel functions. That is, there's a way to actually evaluate elements of the sets via this. I'm not going to go into that in depth, but if you're thinking about things like regression problems, where you want to be able to very flexibly model classes of functions, using kernel methods helps with that as well. So the kernels give us a very compact tool for doing lots of things that we often want to do. Now, that may excite you, or it may not. If it doesn't excite you and you're like, so what? Okay, well, here's a so what. Okay, as a practical matter, why do we use kernels? Well, they give us a very powerful tool for working with complex nonlinear space of functions, right? If I have a huge complicated basis set, I could work with it by mechanically mapping my inputs into that space and then doing what I want to do on them. But if I can use a kernel function to do it all in one go, that's a lot easier. And it turns out in many cases to be very helpful. The real kind of killer app for kernels, though, has been the fact, in machine learning at least, has been the fact that kernels also allow us to non-linearize linear procedures. Now, you know, this is a physical science audience, right? Physical science are all about linearizing things, right? So you're used to thinking about, I take a nonlinear thing and I linearize it. Well, in this case, we want to non-linearize things. And why is that? Well, we have lots and lots of statistical methods and learning methods that are very powerful and can do lots of cool things, but they're linear. So they only work with hyperplanes. And so they're all, you can write all these things down in terms of dot products between vectors in Euclidean space. We would like to apply these to nonlinear settings, but doing that directly can be a complex problem. However, whenever we have a dot product, right, if we replace it with a kernel function, right, we can change the kernel out and we can then use the same method to perform the same technique that we were using before, but now with respect to this alternative basis. And so this gives us a general recipe for taking linear methods and non-linearizing them and generalizing them. And that's been a real killer app because, again, we know a lot about how to do things in the linear world, and it's a lot harder to do them directly in a non-linear way. So just to give some intuition for that, I'll show the example here on the right. This figure was made by my now former postdoc, John Mark Grazielli, who is in the audience. You're going to see more of John Mark's work presently. But so here we have two sets of points, the green and the blue. And a very typical kind of learning problem is to separate those points. So find a decision surface that on one side you're green, on the other side you're blue. Now, obviously, there is such a surface. We can see it. In fact, it's a polynomial. But finding that surface using a linear method is not going to work if you do it directly, right? There's no hyperplane you can pass through those points that's going to separate them cleanly. Now, we could try to find that surface by literally doing some kind of polynomial fit, and there are ways to do it, but it turns out to be a real pain for a whole lot of technical reasons to do it. But an alternative thing to do would be to say, well, maybe if we change the coordinate space of those points and we map them into an alternative basis, we can find another basis where they can be separated by a hyperplane. And indeed, that's what we've done in the bottom panel. So here what we've done is we've applied a transformation of those points. So we're writing them down in terms of an alternative basis set. And then with respect to that basis, there is in fact a separating hyperplane. And then you can use a simple, any linear learning method you like to find a plane that separates those points. And so the beauty of doing it that way is that I can use the methods that I already know and love that are linear to solve a nonlinear problem. And that has been really the killer app for kernels in a machine learning context. Now, there's another cool thing about kernels, and I'll demonstrate that later in the talk, and that is that kernels can be constructed to exploit particular problem features. And this is something that's particularly nice. In many cases, I can select a kernel that has an direct interpretation in terms of a specific choice of feed, okay, a specific feature vector. And that's something that I think is actually particularly useful for intelligible machine learning, right? We want to be able to not only discover patterns, but we want to know what they are. The fact that we can choose that basis set is powerful as well. So this is sort of a very abstract set of ideas, and we could go on with that, but I'm not going to. And what I want to do to make it a little more concrete is talk about some ways that we actually have been using this in our work. And so I'm going to start with a really simple application, and that's to a common problem, which is to characterize dynamics on a complex free energy surface. So here's a little example. This is again also John Mark's handiwork, where we have an ugly little potential, and we have a system that's evolving with respect to that potential. And so, if we have a case of this kind, right, how would we like to simplify that? Well, it would be convenient to be able to simplify it by taking all the basins in that space, cutting them apart, so segmenting that free energy surface. And then we could model or approximately model the space by just looking at the transition rates between the wells and the dwell times within the wells. And especially if we have a very complex problem, this could achieve a lot of simplification, can be quite useful. And there's a lot of other techniques that then can exploit those kinds of ideas, things like milestoning and the like. The problem, though, is that usually we do not have some kind of cute two-dimensional thing where we can see the space. Usually, in fact, we're going to have some very high-dimensional case where we don't have direct access to the energy surface. And also, in practice, what we can only see are the trajectories that are produced. So we might have, say, a molecular dynamic simulation, a simulation like the little balls running around. We can see where they bend, so we can see little traces of them, the particles, but we don't actually get to see the surface. And so we would like to be able to infer what's going on just from seeing those traces. And so this raises a question. If all I can see is the traces, how do I actually find the barriers between those different parts of the space? And what do we do if those barriers are, to use a term of art, ugly? Okay, so there's no guarantee that they're going to be nice linear things. They may in fact be quite complex. And so the question is, well, how can we do this? And so this is something that Juan Adrishoy and I worked on. Again, John Mark did the yeoman's work on this project. This is some stuff we published in JCP a couple of years ago. And so this is sort of a general technique that we developed to do that, and which machine learning is like a piece of that puzzle. So we start out with, actually, before we can do the machine learning part, we have to actually be able to trace out trajectories in space. This is, of course, a well-known problem, and there are lots of imperfect solutions. But for those who aren't familiar with it, so let's start with the idea we have our potential, like, for instance, the case in the upper left, which we just looked at, and it may be quite complex. The question then is, okay, how do we first of all trace out how do we find the basins? How do we trace out trajectories here? And so, well, the naive thing to do is to say, well, let's just simulate some trajectories. We dump them somewhere and we run them, and you could do that. But the problem is that, especially if you're dealing with something like, say, a protein, it's very complex. You probably can't afford to run it long enough to look at the whole space. And so if you do unassisted trajectory sampling, you tend to get stuff like bottom left, where you found a few wells and not much else. Now there's another very natural thing to do here that's widely done, and that is to say, well, let's just turn up the temperature. Let's run it at high temperature, and then that does get you around the space a lot more readily, and you get something like the top right panel here. And it'll work. The problem with that, though, is that now we spend a lot of time, first of all, in very high energy parts of the system. And the second problem is now we've blurred our wells, so it's very hard now to tell where one ends and one begins. So there's a lot of questions about, okay, how do we get around that? And there are various approaches, things like metadynamics, for instance. So we introduced one particular strategy, and it's based on the idea of mutually repulsive trajectories. What we do is we launch a trajectory, and then sort of like the little tree here you see sort of superimposed on this potential on the top left. What we do is that we have the trajectories that we've launched repel each other. So we create fictitious forces that push them apart. And then every so often, those trajectories spawn new trajectories which repel, and those spawn new ones which repel, and so forth. And by that means, what we can do is we can kind of force the trajectories to not cluster in particular wells, and they will kind of meander out. But on the other hand, once they're alone within a well, they don't experience a lot of forcing, and so they trace the wells out in a much more narrow way. And so we use this mutual repulsive strategy. It turns out to be quite good at finding wells, and it generates relatively well-separated trajectories like the ones you see down in the bottom right. Okay, so that's great. Now we have a bunch of trajectories. Now what? Okay, well, we can use some simple clustering methods to find, where the basins are. That part's not so difficult. But then the question we're really interested in is, okay, how do we cut the space apart? How do we find the barriers between in a very flexible way? And so one approach that one can use, and the one that we did in this work, is we used kernelized SVM. So SVM is a support vector machine. It's a tool that, again, is originally a linear learning algorithm. And what it does when used as a classifier is it finds maximum margin hyperplanes. So that is, it takes two sets and it finds a hyperplane between the two sets that maximizes the margin between the two sets, right? So it sort of cuts in between. And so that's a very natural way to cut sets when you're trying to segment a free energy surface. And so indeed, it's also quite flexible. We kernelize it. We can do this now in a nonlinear way, and we can work in high-dimensional spaces without any particular problem. We can deal with boundaries that are really nonlinear, and it's quite straightforward. And so in this case, what we did is we trained classifiers with each pair of adjacent basins, and indeed we get a segmentation like the one you see here at the top right. And so indeed we're able to find the barriers between these different wells. And now that we've figured out what parts of the phase space should be, or sorry, the free energy surface should be associated with particular basins, right now we can use like Markovian models or things of that kind to create a simplified representation of the system. We can use milestoning or other techniques to look at the transition times between those basins and all kinds of good stuff. Okay, so this is just a...
Excuse me, Carter. Sorry to interrupt. Do you have to identify the basins in advance, or does it find them for you? Is this supervised or unsupervised?
Yeah, so there are different ways to do it. There are ways to run SVM in an unsupervised kind of way and do it in one go. What we do, however, is we start out by using a clustering approach to first of all identify the different basins. And there's some natural reasons to do that. And one is that you can relatively easily figure out where your low energy areas are. And so you have a lot of prior information about at least where the middle of those things are. Right. And so it's natural to use other kinds of clustering methods, and there's a ton of things you can use to identify kind of where your basins are, and then we use SVM to cut the space. So we do it in 2 stages, but you can do it in other ways too.
Yeah, thank you.
One way to do it. Yeah. So there's a lot of different variations certainly on this sort of theme. But this gives you a flavor of a way that you can use kernels to help solve this kind of problem in a very flexible manner. So that's one context where we might want to take some kind of space and segment it. Now, another kind of context where we would want to do this is we're looking at phase spaces when we want to separate trajectories, particularly trajectories that may be destined for different kinds of outcomes. And so to switch to another application that we did, and this is work that we published in JCIM last year. Again, John Mark is key on this, and also Siswata Roy in the first lab also helped us with some simulation. I'm going to show you that in a minute. But what we were interested here in is looking at how to use kernel methods to assist with a problem that's sort of a classical kind of mechanics problem. And so here's, to get it just to what this is about, let me start with a simplified example. So I'm going to start with one of the simplest systems that we all know and love, right? The double well potential. And so you can see in the upper right, here's my potential. Because, here I'm going to apply this to molecular things, we're going to label one of these wells R for reactant and one for P for product. That's how you know it's chemistry. Okay, if I didn't have R and P, then it would be something else for a different audience. Okay, so we might imagine that we start out with some reactants. If we perturb them, right, that maybe a little bit, maybe they jiggle around a bit, but they don't wind up fundamentally switching to the product state. If we hit them harder, they may in fact transition to the product state. And so if we consider a little system like that of that type, and we make it dissipative, if we were to simulate trajectories from it, we might get something like these trajectories here in orange and blue. So here we've labeled the trajectory in orange if it winds up back in the reactant bin, and blue if it winds up in the product bin. Okay, this is a very, very trivial one-dimensional example. So we've got the velocity and the y-axis, position on the x-axis. And if we wanted to characterize the behavior of the system without having to manually simulate every trajectory, right, how would we do it? Well, if this is, classical mechanics, right, what would we do? You've got a phase space, right? So we would solve for the separatrices, right, that separate the trajectories that are bound for one state from those bound for another one. So we would separate the basins of traction of the states by the separatrices. Okay, and that's a beautiful thing to do. The problem is that does not really generalize to more complicated systems, right? So if we have a more realistic setting, we're going to have many, many dimensions. So the phase space is going to be too large to treat in that fashion, and we're not going to have direct access to the potential in many cases. It's going to be too hard to work with. So what we're really going to have to do is simulate some trajectories and look at where they go, and then infer the separatrices. And so again, this is a problem where we want to cut something apart. But now we're separating trajectories that are bound for one outcome from those that are bound to another one. So if we're desperately seeking separatrices, how can we do it? And well, you can guess where we're going, right? One weird trick for finding separatrices, namely the kernel trick. So this is another case where actually SVMs are a fairly natural way to go. And so we're again going to have nonlinear boundaries between sets of, in this case, of trajectories. Namely those that separating those that go to one state from those that go to another one. And so we can apply the same kinds of methods I talked about before to do this. And it has all of the same advantages. So we can do this in high dimensional space. The separating surface can be very nonlinear in the input space. And we don't have to know the shape of that separator a priori, right? We can get this from the SVM solution. To see how this plays out, let's go back to our example and sort of show what it looks like if you do this. So here what we've done is we've taken our simulated trajectories and we've then used them to train an SVM to classify where the trajectories are going. So the cells that are in yellow or orange are the ones that are predicted to converge back to the reactant state. The ones in blue back to the product state. And for how do we find the separatrices? Well, one way we can get a sense of where the separatrices are is to look for the particular points in that space that are right on the edge of going the other direction, right? So if you think about trajectories, I've got some trajectories that are they're headed for the product state, but if you nudge them just a little bit, they instead could go to the reactant state or vice versa. Okay, and those are going to correspond to in the SVM solution we call support vectors. So support vector machine, named after the thing called support vectors. And what are they? Well, in the feature space, those are the points that are kind of on the convex hull of the point set, one of the 2 point sets, and sort of facing the other one, if you like. These are points that are on the margin. And so they are also the hardest points to classify, right? So these are the ones that are really, it could go one way, it could go the other. And so since our solution is based on the support vectors, we can easily pull them out. And so here are these black dots are the support vectors. And what we can do to figure out where the separatrices are is we can do several things. One thing we can do is we can look for trajectories that contribute many support vectors, right? So these are trajectories that are hugging that separatrix. And we can look at also ones that are facing, we can look at the facing points on the other side, and we can split the difference in the input space, and that gives us an approximation to the pre-image of that separatrix. So here what I've shown in cyan are those approximate the trajectories that are highlighted, and those are tracing out approximately where the separatrices are. And so once we have that information, we could do things like linearize across the boundary to get a sense of, well, what is the local variables that are making a difference, whether you go one way or another, or do lots of other kinds of analyses. So to give you a sense of what you can do with that, I'll show you an example, a more realistic example, and then a couple of the things that we did with that. And so the example that I'm going to show is photodissociation of acetaldehyde. So this is a widely encountered, mildly toxic carcinogen. Most of you know it as the decomposition, or have experienced it as the decomposition product of ethanol by alcohol dehydrogenase. It's also the thing that slug poison turns into when slugs eat it. So in case you ever wondered how you kill a slug, there you go. Unfortunately, it's a common industrial pollutant as well. So in the atmosphere, it can get hit by UV light, and it can dissociate into various nasty things, including CO groups that can become carbon monoxide, and aldehyde groups and other exciting things. And so indeed, the sort of top right, it shows the typical sort of settings. So if you have acetaldehyde in the reactant state and you hit it with a laser or a high energy photon, then in some cases it'll just jiggle around and relax back and stay what it is. But in some cases you'll lose the H7 proton there. In some cases you will lose that carbon-carbon bond. And in some cases you lose both those things. And so one of the reasons that this is an interesting system to study for people who are into reaction trajectories is that it can really go any of these different directions. And it's figuring out what leads it to do one thing versus another is quite subtle. And so this is a natural case to try to characterize, okay, what are the things that are going to lead it to go one way or another. So to look at this, what we did is we used I did analysis of some ab initio MD simulations that were produced by Siswata Roy in the first year group, who brought us this interesting data. So he had 387 trajectories, which is 2 picoseconds long. And what we did was we took points along those trajectories, and we trained an SVM, a kernelized SVM classifier, to predict the final state, that is where the thing's going to wind up based on where it is. So at a given moment, we can see all the coordinates. We see all that we're representing here in terms of distance between the atoms. We have the momenta, and the question is, okay, where is it going to wind up? And so once we have that classifier, we can do a lot of things with it. Now, we're not going to look at the separatrices because they're really high dimensional and hard to look at. But now that we know where they are, we can do a lot of other analyses as well. And so, for instance, one kind of question that we can use this to help answer is, what are the dynamics that commit a trajectory to a particular product state, and when do they happen? And so to get to the latter question, there's a really easy trick we can use to get some insight into it. And that is, what we can do is we can look at your ability to predict where the trajectory is going to go as a function of how far along you are. So obviously, right, if you let the trajectory run for a very long time, eventually you'll find out where it goes because it goes there, right? So it isn't very impressive to be able to predict what product state you wind up in, given that you waited, a very, very long time. In other cases, you may be able to predict it very early. So the key insight is that if your particular dynamics that take place that are going to commit that trajectory to a particular outcome, right? If those dynamics are still unfolding, you're going to have a very hard time predicting where it's going to go. But if those things have already unfolded, now you know. And so by looking at how accurately you can predict the outcome as a function of time, you can get a sense of the time scale of critical dynamics in this system. And so what I've shown here in this plot is the simulation time in femtoseconds and the fraction of trajectories that are accurately predicted as a function of where they wind up. And so we can see that for some things like the state P1, which is this point here where you just lose the carbon-carbon bond and you form this. So the observation that we're just making is that this transition that we get after about 5 to 700 femtoseconds, right? We now have a sense of whether we're going to P2 state or not, right? So for some things we can immediately tell, and that suggests that the key degrees of freedom are already locked in. In other ones, it takes longer. And so that gives us a sense of what's the time scale of those critical dynamics, right? And that's useful if you want to, for instance, do more directed simulation studies. That tells you, okay, this is where you should be spending your compute time. But it also, of course, can be useful for guiding theory creation, right? Because if we know there's certain time scales that are characteristic of particular processes, and we know that that the thing we're interested in is happening much faster than that, then we know that, okay, that's not the mechanism that's key. And so this is a fast way we can get some insight into that type of question.
Carter, looking at P1 there, it looks like it's just a certain vibration that then sort of goes all the way to some product state. Is that right? It looks like if you start a certain vibration, you have a 90% chance almost immediately.
That's right. Well, yeah, essentially that's right. It's with certain initial conditions, it's extremely likely that you're going to wind up there. So so that's one where the conditions that are going to set you up to have that carbon-carbon break look like there, and particularly for that to be the unique outcome, are quite distinctive, right? So there's something about that initial, that interaction that's driving it very rapidly, which is less true of the other things.
Thanks.
So that's, I can't tell you exactly what it is, although, there are things I can tell you. So let's advance to actually I think it's a little related to that. So we haven't looked at that specific question, and indeed, that requires going into other stuff that's beyond what we've done on that system for sure. But another thing that we would now be interested in is saying, okay, well, what can we say about in those early periods? What does seem to push you toward one outcome versus another one? And to look at that, it can be useful to again use the classifier another way, and that is to tell us which of the states are high decision value states. So these are the opposite of support vectors. These are the states where you immediately know where it's going to go. And these are kind of paradigmatic examples of trajectories that can go one way or another one. And so if we take out these high decision value states that are early in the trajectory, and then we look at what their degrees of freedom are. So we look at their different properties, and we can look at which ones separate those that are bound for one state versus another one to get a sense of what are the kind of key degrees of freedom. And so this is an example of that. So what we've done here is we split things that are going to state P1 versus state P3. And we've just done a PCA on those points. They're labeled here by what their destination state is. So the blue is P1, yellow is P3. And then what we've done is projected into that space different properties of the inputs. And so here in red are the things that are interatomic distances. In blue, we have normalized velocity dot products and correlations of velocities. And then we have momenta in purple. And so we can see immediately there's some kinds of characteristics that, at least for this distinction, are just not very informative. So for instance, some which you might think might be. So total kinetic energy is actually not key, for instance. And neither is the carbon oxygen correlation, for instance. On the other hand, there are things that do seem to matter quite a lot. So the momentum of that H7 proton is quite critical, which makes a lot of sense. There are also correlations between the carbon proton velocities turn out to be quite diagnostic in this particular case. So again, what this is doing. It's not telling us all the answers, but what it's doing is it's allowing us to zoom in on particular degrees of freedom that seem to be determinative of where things go. Once you know which degrees of freedom are important, now you can do other more targeted analyses to try to understand what's going on. Whereas otherwise, all you'd have is a ton of trajectories and you wouldn't know where to begin. And so this is, I think, one of the really useful ways we could use machine learning methods in this kind of exploratory mode to help guide theory by sort of saying, okay, what are the timescales that matter? What are the degrees of freedom that matter? Sifting through all that complexity down to something simpler. So then we can try to understand, okay, what's happening in those particular places. Of course, another way, or another thing that we can do with kernels that will help us in that kind of task, as I alluded to earlier on, is we can use them to when we have an idea of what some of the degrees of freedom might be that might matter, to then zoom in on those degrees of freedom per se. And so one of the nice things I think about kernel methods is that they do allow us to build feature spaces that are informative for our problem without having to pick every single one manually. So it's a nice compromise between, some methods that do their own feature engineering, like deep learning, and methods where you're manually curating every single thing. And so how are we doing this? Well, of course, we remember, right, we can think about the kernel as just performing a dot product in some particular feature space. So by choosing the kernel function, right, choosing the space. And those don't have to be, simple things like polynomials. We could, in fact, have feature spaces that embody characteristics of more complex objects. So take objects that are near and dear to my own heart. We could be looking at graphs. So an example would be feature spaces for labeled graphs. Labeled graphs arise quite often, and for instance, in the context I'm going to talk about today, when we study protein structures. So protein structures are extremely complex. We often need ways to simplify them, coarsen them, so we can handle what's going on. And one of the ways to do that is to use what we call protein structure networks to summarize them. And so we take, say, a trajectory that we've acquired from molecular dynamics, or a fixed structure that we got from NMR or from other methods, X-ray crystallography. We could take the atomic positions of the atoms in different chemical groups. We can aggregate those into vertices. There are different ways of doing that. And then we can look at which groups are interacting and use this to construct a network, the one you see in this case. And so then if we have, say, an MD trajectory, we may have a whole series of networks, and we're interested in identifying things like when is the protein in one state versus another one based on similarities in the networks. And the question then is, well, how can we construct a kernel that effectively picks up similarities in the structure of those graphs? Well, an easy thing we could do is we could just look at, which edges do you have in common? And indeed, there are Hamming distance space methods to do that. But it may turn out that there are particular substructures of higher order that matter as well. So there could be particular shared partner configurations, for instance, that matter, particular triangles that matter, or other things, right, that make a difference. And the question is, well, how could we identify those, but without having to enumerate them all? Because there are too many for us to do that. And so to see how we can do that and construct a kernel that manages to do that automatically, I'll show you an example. And to see how we do it, we have to take an excursion that at first blush seems like it's something unrelated, and that is looking at logic learning. It turns out, in fact, it is related. So, logic learning is the problem of what it sounds like. It's applying learning methods to logical vectors. So I have a series of propositions, and I'm interested in functions of those propositions. And there are families of kernels that have been developed to do exactly that, which are called Boolean functional kernels. So their inputs are vectors of zeros and ones, which in the original context means true or false, and then those are the feature spaces are based on those. So an excellent example of this that we're going to wind up using is something called the disjunctive normal form kernel, which is developed by Kowalczyk and colleagues in the early 2000s. So the feature space of this kernel is the set of all possible conjunctions on a set of propositions. So you give me a set of propositions, and the feature space is, do I have, is X1 true? Is X1 and X2 true? Is X1, X2, X3 true? And so forth. all of the possible cases, which is, of course, a lot of propositions. So you wouldn't want to have to manually enter them. But the beauty of it is that we can, in fact, consider that feature space and do operations with the feature space without having to enumerate them. And the trick that's used is what's illustrated here on the right. So we have, again, our kernel function, and what this DNF kernel does is it says, and remember, in the kernel function, we're comparing two things. So we're doing an inner product between two different vectors here, x and y. And so the first, you can see the sort of first terms, set of terms I've grouped, what they have in is they count all of the specific individual propositions that x and y have in common. So is x1 true for x and for y, how about x2 and so forth. And so we're going to take all of those and we're going to weight them all by a value epsilon. And then we're going to count all of the pairs of propositions that they both have in common. And we're going to weight that by epsilon squared. Now, why are we doing that? Well, the intuition is epsilon, we're going to set epsilon less or equal to 0. And so the idea is that because there are more and more and more and more conjunctions, right? As we get more and more of them, we want to be able to downweight those things so they don't overwhelm the solution. And so we're going to weight them down exponentially by the conjunction lengths. So we weight those by epsilon squared. Now we consider all the sets of 3 and all sets of 4 and 5 and so forth, right? And of course, the combinatorics explode. You would not want to try to do this manually. But we can, it turns out, do them tacitly just by using some standard series tricks. So if we write out that expression above, it turns out it reduces to this really nice little polynomial. So it just winds up being 1 plus epsilon to the dot product of x and y. That is how many propositions x and y have in common, minus 1. And so that's an example, right, where we can have a very, very complicated operation that we would not do manually. But with the kernel, right, we can do that operation tacitly. So it's enormous computational savings. I'll also observe that this kernel has a hyperparameter, right? So we have this epsilon, and what that's doing is it's regularizing the solution. So when that hyperparameter is close to 0, then that says, you know, these higher order conjunctions are not that important. What really matters is just, you know, do you have individual propositions in common? When that parameter is closer to 1, that says, hey, these higher order things really matter. It's not just do you have particular individual things in common, but do you have larger sets? And so so this is quite a nice, flexible kernel. There's lots of cool stuff you can do with it. Now, at first blush, what does it have to do with graphs? Well, to see why it has something to do with graphs, we have to recognize that there is a duality between logical functions or Boolean functions and networks vis-a-vis the adjacency matrix representation of network. So if I have here it's a very small network, like shown here at the top right, I can represent that network, right, via its adjacency matrix. And so in this case, it's very small. It only has 3 nodes, 3 edges. And we can represent that by the 3 edge variables, x1, x2, and x3, expressing the connections between I and j, I and k, and j and k. And so we can take the vectorized representation of that adjacency matrix and apply a DNF kernel to it. And if we do that, what we are implicitly doing is we are counting labeled subgraphs. And in particular, we're looking, when we take the kernel function, we're looking at whether or not x and y have particular subgraphs in common. And indeed, there are a lot of subgraphs, right? We can see even with the 3-node network, right, there are 7 of them. I've enumerated them all here on the right. And so we can tacitly look to see whether or not the network has this thing in common, the other thing in common, without having to enumerate all that stuff, because there's no way we could do it in practice. So to give, again, make it concrete, like what are we doing when we take an inner product and graph space with this kernel? Well, this is what we're doing, right? So if I've got K operating on these 2 graphs, so this 2-path here on the left in purple, and this triangle in orange, well, what am I doing? Tacitly, I'm looking at all the possible subgraphs I could have. I'm for each one saying, is it present or absent? If they are both present, I'm going to weight that by the appropriate multiple of epsilon, and I add it all up. Okay, and so we indeed wind up with doing exactly what we would expect. So it's a very natural thing to want to do. We couldn't do it manually, but with this kernel, we can do it automatically. And so this gives us a very elegant way to do something that would otherwise be very hard. Okay, so now that we can do it, what are we going to do with it? And I'm going to show an illustration from some current work that we're doing in conjunction with the Martin Lab, looking at the dynamics of SARS-CoV-2 Mpro, so the main protease of SARS-CoV-2. So as many of you know, when viruses infest your cells, in order to do their dastardly deeds, they have to use protein to do it. So they trick your cells into making these proteins. And typically they do that by getting your cells to express one very, very long amino acid chain polyprotein that then has to be cleaved individual parts in order for the virus to do its villainous work. And Mpro is the thing that does the cleaving. So if you can inhibit Mpro, you can stop viral replication. It's an important clinical target. What we're interested in particular is understanding how the mutational landscape of Mpro affects its dynamics. And so what we've been doing is collecting all of the clinically observed variants of Mpro that have been found. There's folks at GSAID keep a database of this, particular shout out to TJ Cross and Gemma Takahashi and the Martin Lab that have been doing yeoman's work to collect all of the sequences that arise and find the unique ones for us. So for what I'm going to show today, this is all the ones that existed up till May. So this is wild type and 78 mutants. We're now up to 400 some mutants. So it's really quite a large collection. And then what we do is once we have those variants, we model them. So we use a combination of homology modeling and atomistic molecular dynamics to model the structures. We then put them in explicit solvent, and we obtain trajectories both in the monomeric form and in the more biologically relevant dimeric form for each of these cases. So we wind up with a lot of MD trajectories. So we want to understand. We've done a lot of things with them, but what I'm going to talk about now is going to be focused on some stuff that we're doing with the active sites here. So here you can see the Mpro active site. We have a catalytic cysteine and histidine. We're going to be looking at these by using protein structure networks. In particular, we're going to work with something called an active site network, where we take all of the chemical groups associated with those catalytic residues, and then we take all of the other groups that are in contact with them, and we build a sort of local network around that. And we can look at how those change across variants and through time to try to understand things about how the dynamics of the Mpro active site are related to other factors. And so there's a lot of stuff we can look at. And we started with just looking at characteristics that we knew from prior work were potentially important. And so things that were kind of done on a kind of custom basis. So to give you a flavor for what this is like, I'll show you an example. So this is a visualization created by Liz Diesner in my lab. You can see on the far right, that's Mpro, the Mpro dimer with the active site highlighted. In the middle, you can see a zoom in on what the active site is like, and then you can see the associated active site network. So those yellow nodes are the cysteine, and the blue nodes are the histidine, and any two nodes are in contact if they are within 10%, 110% of the respective, some of the respective van der Waals radii, so they're in contact. And the examples I've shown are organized around something we call constraint. So this is a network analytic measure that looks at the extent to which those active site residues, so the catalytic residues are hemmed in with lots of other contacts around them. So they're not very flexible. And so we can look at that across the variants. So the far left here shows the means and autocorrelation adjusted 95% confidence intervals for the means from the simulations for the dimeric form. So there are two chains in each dimer, and each row is a variant. The green one is the wild type. And you can see that there's a lot of red here, and what that's showing is that there is some selection pressure toward getting variants that have less constrained active sites, so they're somewhat looser. And we've gotten some insights and things like that, and that's a case where we thought in advance about what are the features that matter. We looked at them, and that's a great thing to do. And it showed us that there was a lot of variation between these things. But what about other kinds of variation where we may not know what it is that's important? It's very hard to figure out how you discover that, and that's something where ML can help. Right? So we'd like some sense of like how all of these different active site conformations differ. We want to be able to answer questions like whether or not the monomer conformations differ radically from dimer conformations. And we know there's a huge difference in activity between the dimer and the monomer. Is that because of the active site conformation, or is it because of something other than the active site involving, say, substrate interactions outside the active site? How big are the differences by variant is an important question. And also, can we identify experimental observables for further studies? So specific interactions that we can tune into using, say, MR methods. And so to do that, one of the things that we did is we did an exploratory analysis using kernelized principal component analysis. So PCA you're probably all familiar with, right? We take Euclidean space, We find an orthogonal basis in the space. It's the first dimension explains the most variance, the second dimension explains the next highest variance, and so forth. Well, now what we want to do that is do that in our feature space, and we're going to do that in that graph feature space. So we're going to find the dimensions that explain the most commonality in which chemical groups are interacting. And so we can do that here. We take all of our networks, we take a subsample of those, we run it. There are about 9,000 that we used in this analysis, so there's no shortage of them. And what we do is we now look at what's going on in that space of the kernel space, in that space of shared subgraphs. And when we do that, and we pull out the first two principal components, we get a lot of insights immediately. One of the things we see is that there are really kind of two classes of trajectories here, these two clusters that are quite visible. I've shown the centers of those two clusters in feature space, so the two networks that are most like the others. And if we ask, well, what separates those clusters? Well, it turns out there's like one weird thing that does it, or maybe not that weird, but one thing. If you look closely at the figure, you'll see that one cluster is dominated by circles, the other by triangles. Well, the circles are all examples of conformations where there is a tie between a cysteine and a histidine node. And so if you look at, in fact, at that blue case, you can see that I've circled one of those edges, and indeed that is one of those contacts. And you see the red case does not have it, and that's because that's an open conformation without it. So that one thing is that, are those two residues in contact or not, is the big differentiator between conformations in this case. And then the other two things that seem to differentiate them overall is the mean degree. So how many chemical groups are different groups in contact with, and the size of the ASN, which is shown here as a gradient, and that's just how many other nodes are interacting with the active site. Once we have this sort of quantitative measure, we can do other things as well. So we can even just use a simple ANOVA to say something about variance explain. So a big question, is it a monomer dimer difference that matters? And the answer, no. Only about 3% of the scores are explainable by monomer dimer differences or even dimer chains. What about the mutation? So what about the variance? About 10% can be accounted for just by that. If you take the interaction of those two, it adds about 23% more. So about a third of the variance overall can be explained by what type of variant it is. So there is definitely quantitative variation there, although we find there's no one dimension that aligns exactly with any of those distinctions. So this gives us already some really useful insights, and it rules out immediately one really natural hypothesis of what might be going on, which is it's just monomer dimer differences. But we can go a little further, and we can use these components to actually figure out what are the specific interactions that characterize these differences in conformations that we see. And so to do that, what we do is we look at edges that are high contrast edges, okay, that really demarcate things that score highly on a component and things that score low on the component. And so here I visualize that on the active site itself. So we've gone back into the space of the molecule itself. And so you can see that some of these chemical groups are connected by red lines, some are blue lines. And what the principal component is doing is it's contrasting interactions marked by the blue lines with the ones marked by the red lines. And so this gives us a more interpretable way to understand what are the connections that seem to matter. And so we can see that that first principal component, for instance, is really contrasting conformations where that histidine 41 is in contact with C145. Those are the two catalytic residues. But beyond that, that there are a bunch of other interactions that are important as well. So in one of those conformations, for instance, where the two are in contact, they're reinforced by mutual contact with histidine 164 backbone, and that's helping to stabilize them. In the opposite case, that histidine is wandered over, and it's now being cradled between arginine 40 and the aspartic acid 187 that are forming a salt bridge. And then there's a kind of tyrosine, the OH group on that tyrosine is kind of like keeping its finger on the top. So there's a sort of cradle that captures the histidine at the other side. And so this gives us a more concrete sense of what the difference is. If we move to the second principal component, that's a different contrast. It does also involve that cis-his interaction, because we've seen that's an important one. But it has a number of other things going on as well. So for instance, this is a contrast where not only are the cis-his in contact, but they're also interacting with this proline 39, another cysteine cis 44, and now the ring on H164, so a different part of that. whereas in the cradle conformation, which still does show up, there's an involvement with another cysteine C85. And so when this gives us a much richer sense of, as this thing is moving around, like what are the conformations that account for most of the variation? What are the key different states it's occupying? And then being able to translate that back to specific interactions in the proteins. That gives, and not just even at the residue level, but at the level of particular parts of the residue. That gives us targets that we can then go in with NMR or other methods and actually try to measure sort of how often are these contacts present, when is it in one subconformation versus another one, and that's something that we can then use for follow-up. So if we just tried to stare at 6,000 different active site conformations, we were not going to see that. We were not going to be able to find that out. But by using appropriately tuned kernel method, we can identify those kinds of details that then are a good follow-up for further work.
Right. Sorry, Carter. Oh, yep. I just noticed that we're almost at 3 o'clock, so we're
well. Good, because I am about at the end here. So, so we lost a little time on our technical issue, but I'm closing it up anyway. So it's a good time for that. So obviously we're all here because well, there's a massive surge of progress, interest in machine learning everywhere, including physical sciences. I do think there's a lot of opportunity, but there is a lot of hype, and I think it's important for us to think carefully about trying to do analyses that actually give us scientific insight. It's impossible to do this work without getting drawn in the meta debate between the people who see machine learning as a way of implementing non-parametric statistics and machine learning as a replacement for theory, or maybe like thinking ever. You just turn the magic crank and numbers pop out, and the problem with that latter point of view is it leads to the difficulty that you get machine learning where the machine learns and you don't, and I don't think that that's really what we want to achieve. So I'm an unabashed advocate of kind of intelligible problem-driven machine learning rather than just black boxes. I mean, there's a place for black boxes. Like sometimes you want your deep learning for your big data, and it has its use, but I do think science is historically progressed when we try to think carefully about what's going on, and we use these new tools to enable thoughtful consideration of problems. And I think there's lots of opportunities to do that. Kernel methods, I think, are often really well adapted to that kind of challenge. They're certainly not the universal solution to all problems by any measure, but they are often a natural fit for problems of physical sciences, both because there's a natural match between solution geometry and the kind of answers you want to get, and also because there are some really nice technical connections between kernel methods and other ideas that are very well understood in the physical sciences, and I think there's a lot of room for advance as a result. So to close, let me again thank folks who did the work, particularly Jean-Marc, and now San Jose State, and the chemistry faculty there. Liz Diesner and my group also worked on some of the stuff I showed today. Then Yuan, Saswata, Rachel, collaborators, and then in the Martin Lab, particularly TJ Cross and Gemma Takahashi, and some of the stuff that I talked about today. So thanks to all of those folks for their contributions, and thanks to all of you for tuning in. And if there is any time people want to stick around, I'm happy to answer questions.
There is one question from user. Is there a course or resource you recommend for mastering kernel methods? And Jean-Marc Grazioli sent a link to a book. It's on the chat for those of you who are interested.
Yeah, I don't know. I can't see which was the way my stuff is set up. What the which book he recommended, but yeah, I mean, there's a I don't have a very specific thing, except there's a book called Learning with Kernels that I find quite nice. It's oh gosh, Smolin. I always forget the other author, but anyway, I particularly like that book, but Google is an amazing thing these days. I don't. My usual, I mean, actually, I'll tell you how I think you should learn it, but no, you're not going to like my answer. I think you should learn it by going and implementing it yourself. This is how I learn everything, is I go write my own code. And so, find a good problem and then start writing code to implement your own kernel methods, and then you really understand it. But admittedly, that's not an answer that satisfies everyone. It's not the most efficient way to do it, possibly. But I do think you learn a lot by trying to understand how the parts fit together. but anyway, you know, there are a lot of resources out there, but I would recommend that book. It's a good, it's got a lot, it's approachable enough, but it also has a lot of technical meaning.
Yeah, Steve Franklin just posted a link to the book on the chat also.
Yeah, I really like that one.
So do we have questions for Carter from the audience? I have a few. So just before you finished up on the 2 slides back when you were looking at the 3, that one, yeah. So do you remember what fraction of the variance was covered by PC1 and PC2?
Well, that's complicated, right? Because the variance explained is going to be in the feature space. Oh yes. You can't normally. It's not trivial to figure that out, as far as I know.
Right.
So it's not. That's like in a conventional PCA, you're usually, you know, the input space is the feature space, so it's very easy to do that. Right. And here it's not. So you can. I mean, you know, you can look at the magnitudes, the eigenvalues in general, the spectra, right? In a kernel. Typically, the spectra in a kind of kernel PCA are a lot flatter than in a lot of other cases. So
and you don't really know what they mean. Yeah.
But yeah, so that's one of the things. I mean, as soon, you know, you have the usual problem, right? That as soon as you leave the linear world, you know, a lot of things are harder, and you don't have easy answers to a lot of stuff. You know, linear world is a nice place, right? We like to live there, but yeah. So exactly how much variance is explained there, I can't say. What I can say is I can say, okay, out of those dimensions, and I could say that that's certainly the dimension that if I had to pick one dimension in the feature space, right, the first component is the one that has the most variation on it. That we can say.
Wow. And can I ask you, what was the largest matrix diagonalized in the course of this talk?
Now you're getting to the bête noire, right, of kernel methods.
You see where this is going, yeah.
Yeah. Well, it's probably this one, right? So you can see that in this particular case, we had about 6,000 networks, and you know, I did this one just like brute force, right? I mean, standard numerical.
Yeah. So
and this is just, you know, you're doing full, you know, standard diagonalization. So there's nothing really special. Now, if you want to use much larger cases, there's like a whole literature on how you do that.
Yeah.
But this is a you know, so this has been the thing that's challenging about kernel methods, is, you know, there are a couple of things I think that have been challenging and that have enabled the neural network people to get an upper hand. And one is that they get challenging to scale to really large ends. They're often very efficient. But now that you can do it, one way to do it, well, there's a million ways to do approximate diagonalization. That's one of the things you do. And there are a lot of different kinds of approaches. In some cases, you can get away with like stochastic low rank approximations. There are some very clever tricks. If you have the right kind of kernel methods, there are some kind of Fourier analysis tricks where you're able to like very accurately approximate the Gram matrix without ever computing big chunks of it. Mm-hmm. And but not all of those methods generalize to all kernels. So there's a lot more to do, but especially when you get to kernels that are not, you know, non-Gaussian kernels. So I think it's somewhat underexplored. It has, I mean, my own personal sense of it is that it has the challenge that there's a lot that can be done, but it requires thinking about it. It requires math. And an advantage of, I mean, just to be brutal about it, an advantage of like, you know, the kind of the current neural network paradigm is that, you know, people can just slap layers on things and turn cranks and tweak the numerical parts, and they don't have to think too much. And things where you have to think tend to get dominated by things where you don't have to think. It's my observation. So anyway, you know, it's very partisan, in case you're wondering.
You know, this is being recorded, don't you?
You know, like there's, I say controversial things as a matter of course. So I like to make fun of neural networks. This is a known personality flaw. But I'm not. I mean, I'm not that proud. I will use them if I have to. I mean, there's no question that neural network methods have been very successful in many applications, and there are some kinds of problems where right now there's just nothing that can do as well. But at the same time, they often give you solutions that you don't understand and whose quality cannot be assured, and you don't really know what they're doing. And I think in many scientific applications, that's not so ideal. But it depends on what you're trying to achieve, right, and what your problem is. But anyway, yeah. So scaling, going back to your question, right? Being able to scale kernel methods to really large cases is one of these things where there is work on it, but there continue to be a lot of open problems. And I think that's a great area for people who want something really interesting to delve into. Now, another thing, a comment I'll make about that though is that we usually assume that what we need to do is scale the method, right? Like we need to be able to deal with like 1 million observations. But it's not clear that you always need to. In many cases, you don't need that much statistical power. Sometimes you do. There are cases where you do, but there are cases where you really don't. And honestly, a few hundred observations would have more than enough statistical power to learn what you need to learn. And so, in some cases, it seems like smart subsampling would make a lot more sense than than trying to do that. But this is a good question.
Yes. It seems, I mean, what it is once you get past the 1 million mark or so, right, you can just keep pushing the network to get an extra 3% accuracy in a classification problem, right? But the fact that you can do that is what people want in certain applications. But yeah, not if you want to understand what's going on.
Well, and I think where I can say personally, I've observed the differences in really heterogeneous problems. So like text analysis, where you have, so not very physical science, but if you're trying to get at things involving, you're trying to do embeddings that are going to capture the semantics of strings of text, of natural language text. You need so much data to capture the nuance of, even within one domain of natural language, because there are all these different phrases, and also because the neural network's trying to do it by brute force.
Right.
You need, there you really see the difference. Like, you can, I've had examples where I've run things with, 100,000 text strings and it doesn't work. And then 1 million, it works, so you just need an enormous amount of power because your problem is so heterogeneous and you have so many parameters that you're learning.
Right.
But that's a class of problems, and I'm not denying that those are important problems. They are, but many of the problems we work with are not like that. And then you're not really getting, in many cases, you're not necessarily getting a lot for that. For dumping all those data points in. There also is other interesting emerging methods from a different area. A lot of this work has been done in computer science and electrical engineering, actually, but trying to use methods to identify kind of ideal subsets for training. And so high-value data subsets. Some of this has come out of work on trying to value data, like literally put assign a price to data. And so it turns out, and this is also related indirectly to not the work itself is not, but the problem is related to like the use of things like low discrepancy sequences in variance minimization in Monte Carlo quadrature, which some of you will know about. That we know that there are a lot of cases where if you have like carefully chosen data points, you can dramatically improve the performance of an algorithm by lowering the variance of an estimator. And there are things that we're doing in that space in a more machine learning context to say, can I identify minimal data set that gives me essentially as much learning value as the entire data set? And often you can do surprisingly well by choosing a smaller data set. So that's another frontier. I haven't seen that specifically exploited for kernel methods to try to to deal with a small basis set. But there are a lot of interesting ideas out there, I think, waiting to be exploited.
Can I ask one more question, a technical one about the first couple of things you talked about? I mean, especially by using kernel methods, it seems like it really would open the door toward, you know, coarse-grained models, that it actually suggests ideas of which degrees of freedom you need to incorporate and which ones you can forget about. Have you tried any of that where you use a simpler model and see if it behaves the same way as the full calculation?
I agree and no, respectively. So, so yes, like I think that's right. I mean, of course, you know what I mean. Trying to get coarse-grained models was one of the, especially the first application. It's obviously a major application, and you know there is, you know, we're still not the only people to try to do that. Most of the focus there has been on, you know, trying to take your surface, cut it into parts. So if it's like, say, a protein, you have a conformation space, you want to cut that apart, and then you want to have some sort of like, you know, Markovian model where you move from one basin to another one. But it's usually been done by things like, you know, Voronoi tilings in the input space, things like that. And those are, you know, they have their virtues, but they don't, you know, they're not necessarily, there's no guarantee that nature likes to cut things in a way that's clean in your input space. And that's the advantage of doing it this way. But yeah, in principle, what you could then also do is if you, once you have these, you know, once you have these cuts, you could try to see, can you now do a dimension reduction on top of that and then figure out, well, what are the really key inputs that are actually driving it. The other thing is they don't necessarily have to be uniform, right? So you could imagine that in certain parts of that space, there are a few degrees of freedom that are critical, and other ones may be critical in other parts of the space. And if you understood that, that in and of itself might lead to kind of new ways of thinking about modeling the problem. So I mean, it seems like a really excellent direction. It's not something that we've tried, but you know, I think it's quite open. But I completely agree that that's useful. I mean, I think in general, right, we want to try to reduce the numbers of degrees of freedom, not expand them. So I kind of find myself using these sorts of things, like as the example at the end showed, is as a way to cast a wide net, but then, you know, kind of go back and look at what I caught that was important, and then say, okay, can we now that we know sort of what's important, can we now just focus on those things? And I think that's a method, that's an approach that's likely to prove productive more generally, but of course we'll see what happens.
Yeah, and I guess it also has the potential to be systematic in the sense that you find the most important stuff, have a model that captures that, and then you can actually say what the next correction should be. Yeah, test that out, and that would be wonderful, right? That's very hard with these multi-scale problems.
Absolutely. Yeah.
Okay, so anyone else want to?
There is one message on the chat box. How does mathematical sociology intersect with machine learning?
Is that, were they asking for a lecture?
No.
How does it intersect with machine learning? Well, I mean, machine learning, right, is it, well, first of all, then you have to define what machine learning is. That's contentious in and of itself, as I hinted at the end. But I mean, I think that, you know, I think that in mathematical sociology, one of the grand challenges that we have is to deal with complex systems of interacting elements, right? And that's one of the threads that unites, like studying proteins and studying humans. At the end of the day, what you have are systems that are heterogeneous. They have multiple parts. The parts interact in complex ways, and the interactions affect the other interactions, right? You have many body effects. And what we do is try to understand those kinds of systems and build models for those systems and build methods for studying them. And so some of their, you know, what we do in that space is, you know, computational modeling. Some of it is more traditional mathematical modeling. But, you know, what machine learning is adding to that class of issues, whether it's in social sciences or physical sciences, is, I think, a tool for a methodological tool for grappling with the complexity of the system, and then ideally kind of finding key patterns that you wouldn't have otherwise been able to pick out that turn out to actually be predictive and important. And once we can identify what those patterns are, we can hope to understand where they come from. We can hope to understand their consequences. We can hope to build generative models for them, just like what, you know, what Karen and I were just discussing. So you know, the differences between these areas are they're not as big as you might think, actually, from a standpoint of theory, right? I mean, as I think Newton, you know, put it, nature is consonant and conformable to herself. You know, the same kinds of problems show up, whether it's in biology or chemistry or the social sciences. The details matter, and the details are different. It's not like you can just blindly do whatever, but there are broad themes that are very similar and challenges that are very similar. And indeed, we can use ideas from one to solve problems together. We just recently published some work on modeling amyloid fibrils using models that were originally developed to study things like friendship and advice seeking among individuals. And yes, we have to modify some things, and it's not just literally you pick it up and you apply it, but we can take all the technical ideas were developed for that application, and we were able to adapt them to actually look at fibril structures. So there are underlying similarities between these different areas. So
there seems to be a lot of excitement with generative pre-trained transformers for natural language processing, like GPT-3 from OpenAI. Do you think that's readily, I mean, could be used in other strategies in physical sciences?
Yeah, it could be. I mean, that gets into stuff that it's not my main thing. So yeah, there's a lot of hype around some of that stuff too, I would also point out, right? If you've ever played with some of those, you know, the GPT things, like, you know, you'll see these things where they show text that's produced by this, and it's like, oh my gosh, the AI is like magic. But then if you generate it yourself, you notice it generates a lot of gibberish, and then sometimes it generates things that are clever. So I mean, to some extent, you know, it's a cute toy, but I mean, you know, there are impressive things behind it. You know, can that be used in other ways to produce things that are more scientifically useful? You know, that logic, possibly so. I mean, in a sense, it's learning a probability model of a kind. A difficulty with some of that stuff, though, is that you don't really know what you're doing. I mean, like you get a model, but you don't necessarily understand the model. And so that can be a challenge. Now, people are working on ways to try to get at that, and you know, there are ideas like using that as an aggressively non-parametric approach to sort of generate a generative model, and then use more intelligible models to study synthetic data from the synthetic model. So you have like kind of 3 stages of modeling where you have the real original data, uses to produce this non-parametric, confusing generative thing, and then you have something more parametric that you use to approximate it, and that has some potential virtues as well. But I don't think at this point anyone really knows like where the limits are. I mean, that's going to be what this generation of folks coming up are going to find out. And I mean, I expect that, like all things, it will not live up to the hype, because nothing does. But I do think useful things will come out. We'll see.
Okay. So I think we'll wrap it up here. I'd like to thank Carter very much for coming, and I think I would say that maybe his talk was the closest to the true spirit of this nexus, which is physical sciences and machine learning. We saw some real chemistry going on there and some real machine learning applied to it. So thank you very much, Carter.
Well, thanks very much for having me. Great to talk to you.
Thank you.
Good job.